Okay, so this is part 2 of protein synthesis. In the last video, we talked about the overall concept of protein synthesis, the overall process, the 2 steps of transcription and translation. So really quickly, we start with DNA. DNA has the instructions for how to build the protein, but we don't go straight from DNA to the protein. Let me tell you why real quick, because DNA is stuck inside the nucleus of a cell. And because DNA can't leave the nucleus of the cell, but they contain the instructions, the protein actually gets built outside of the nucleus. It's still in the cell, but it just gets built outside of the nucleus where the DNA cannot go. So DNA has all the instructions for how to build these proteins, but has no way of actually directly delivering those instructions to the place where the proteins actually get made in your cells. So that's where its little helper buddy, RNA, comes in. And RNA will go in, talk to DNA, get the instructions on what it needs to do, what proteins need to be made, and then take those instructions and deliver them to the places in the cell called ribosomes. There's a ribosome, here's a ribosome. These are all ribosomes. To the places in the cell where those proteins actually get put together. Okay. So before we can make the protein, we have to rewrite the instructions in DNA into a strand of RNA. And that process is called transcription. Then that RNA, the instructions in the RNA, can be used to make the protein, which is called translation. So that's step 1 and that's step 2. Now, let's say something very satisfying about doing this. There we go. All right. So let's say that our strand of DNA is, remember it's made up of GCAT. So let's just say G, C, C, T, A, G. So that's one strand of DNA. DNA is 2-stranded. So there's strand 1. But then there's another strand directly across from it that those guys are linked to. And according to the base pairing rules, G and C always go together, A and T always go together. So across from the G will be a C, across from the C will be a G, across from C will be a G, across from T will be an A, across from A will be a T, and across from G will be a C. That's because of base pairing rules, which we talked about in a previous video. And that's the second strand. So what's going to happen is we're actually going to take one of these 2 strands, let's just say it's this one, and we're going to read this strand, and we're going to rewrite that strand as RNA. So how do we know which of the 2 strands to use? Well, for now, we're not going to get into exactly how that works. Just know that your body knows which one of these 2 it's going to read, and it reads it. And the one that it reads, we call that the template strand. So we're going to read this template strand and we're going to rewrite this or transcribe it with transcription into a strand of RNA. But before we can do that, these two strands, because they're connected like a ladder, we need to go right down the middle between the rungs of the ladder and separate the two sides of the ladder. And this is done by an enzyme We talked about enzymes in a previous video. And remember, enzymes are just molecules that speed up chemical reactions. If you don't know what that is, you got to go back to the enzyme video and refresh on that. So these enzymes, there's an enzyme that comes down here and it actually separates and cuts right between those two and separates strand 1 from strand 2, the template strand. And that enzyme is called, I'm going to put it down here, DNA helicase. Remember, it ends in ACE, it's an enzyme. I mentioned that in a previous video. So this is DNA helicase. This enzyme comes down and it separates these two strands, and then we take the template strand and we're going to rewrite that with transcription into a strand of RNA. But what does that look like? Well, it's not an exact copy. We follow the base pairing rules again. So across from a C, the C is going to be rewritten as a G. This G up here is going to be rewritten as its pair, a C. Same thing with this G, will be rewritten as its pair, the C. And then this A, well, A and T go together, so this should be a T, right? Well, no. Remember, when we were talking about RNA, RNA is made up of G, C, A, and U, where DNA is G, C, A, and T. So when we're rewriting the DNA as a strand of RNA, there is no T. There is a U instead. So instead of rewriting this A as a T like we would if it were in DNA, we're going to rewrite it as a U. This T is going to get rewritten as an A, and this C is going to get rewritten as a G. Okay? So, now what we have is a strand of RNA. This is a strand of RNA. And this strand of RNA now has the instructions on how to build the protein. But how do we decode that? This makes zero sense. It's just random letters in a random order. Or is it? There's actually a code in here. And just like code is supposed to be, it's not supposed to make sense unless you have a decoder, which we have. Let me show you. So here's our decoder. We're going to use this to decode that strand of RNA. But before we can do that, we first have to break up this strand of RNA into groups of 3. Here's a group of 3, GCC, and here's a group of 3, UAG. So once we break them up into groups of 3, these, by the way, are called codons. We got codon 1 and codon 2. So once we break them up into groups of 3, or codons, we're then going to take that and we're going to decode the codons. So let's remember GCC. So what does GCC make? Well, this is a codon wheel. You start in the middle and you work your way out. So we start with G, and then we find the C, and then we find the C. And we find that it makes the amino acid alanine. And you're probably wondering, I'm never going to remember that GCC makes alanine. You don't have to. You'll always have one of these. Just use this. You don't have to memorize all this. Just use this, and that's all you need to do. So alanine is the first codon that gets made, and we're just going to write the first 3 letters. I'm not going to write the whole thing. So alanine is our amino acid. That's the first amino acid in this sequence of amino acids that's going to make a protein. So we're going to call that amino acid 1. Now we do the same thing with the second codon, UAG. So let's see what UAG makes. Let's start here in the middle, U, and then we go to the next one, A, and then the next one is G. And what does that make? Stop. That's a funny name for an amino acid. Why would they name an amino acid stop? Well, it's not an actual amino acid. That is a stopping point. You see, your proteins are only so long. And at a certain point, your DNA tells your body to stop making the amino acids. Stop. And then all the amino acids that got made from the first one until then, those are the ones that are going to make up that protein. Proteins don't go on forever and ever. They're a fixed length. So there's a stop codon that tells it when to stop. And that's what we have here. Now, in reality, there's really no such thing as a 1 amino acid long protein. I just wanted to show you that amino acids were built according to what the codon tells it to build, which is determined by what's in the DNA. And eventually it reaches a stopping point, which is also indicated by a codon, which is also indicated by a sequence in the DNA. So really, this is just a 1 amino acid long protein, but like I said, proteins in real life are hundreds or thousands of amino acids long, and they're all different. And they all vary by the order of the amino acids. How many of those amino acids and what order they're in is what makes the difference between one protein and another, and what job that protein does versus the job that the other proteins do. It could be a protein that makes your hair curly. It could be a protein that does any number. I could go on and on, but I just, what's the point? You get it. So that's the overall process of protein synthesis. DNA has the instructions. It's transcribed into RNA. We break the RNA up into codons. Each codon tells you to make a certain amino acid. We string the amino acids together, and that makes the protein.